We're going to start a lecture here on ATP, sodium, potassium pump, and cardiac action potential. I would just like to preface this lecture. I know this is difficult content, and I expect a significant amount of classroom discussion as well. But I want you to have some familiarity with this before you come to the class. So let's go ahead and get started first with ATP. It stands for adenosine triphosphate. What that means, if you're a chemistry person or microbiology person, you probably understand this, way beyond my level as far as how the phosphoryl groups and the phosphorus comes off of the adenosine. But I can give you the broad overview. Adenosine triphosphate, it is adenosine chemically bonded to 3 phosphoryl groups. Those phosphoryl groups will come off and expend energy, and they can reform and become a different structure when they do come off. And we'll talk about that in a second. So they're kind of like a battery within your body. There's a whole bunch of potential energy that is there and isn't used until that phosphoryl group actually comes off of the adenosine. So it's also referred to as the energy currency in your body. So when your body needs energy, it uses ATP. So think of all the things that require energy in our body. One of them we're going to talk about later in this lecture, the sodium potassium pump, requires energy in order to open these channels. So other things that require energy within your body, active transport. So if there needs to be a substance to move in and out of a cell, ATP is the energy that is used for that. Also, when we look at cellular signaling, the definition of that very plainly is cells communicating to one another for tissue repair and muscle contraction. We need to be able to say, okay, muscle cell next to me and next to you and next to you, almost like a game of telephone, we need to contract. And how does that signal get sent, and how do these channels open? We're going to talk about that a little bit in cardiac action potential today as well. That's done with ATP. So when ATP is used, and I've got a video on this in a little bit, one of the phosphorus comes away, and the gentleman talking about the video gives the analogy that it's like a spring. It's potential energy. When the spring is released, the energy is released. So that adenosine triphosphate is broken down to adenosine diphosphate. Now, a couple things can happen here. It can pick up another phosphorus and become adenosine triphosphate again, or it can lose an additional phosphorus and become adenosine monophosphate. All right. So when we talk about cellular respiration, and that lecture is coming to you next week on Monday, we talk about glycolysis, the Krebs cycle, and further the electron transport system. All right. When that happens, it's creating 38 molecules of adenosine triphosphate. So it's a very, very large return on investment. This whole formula and this whole process, how it breaks down. Also, acid-base balances, and then later on when we get into pharmacology, we actually give adenosine to help slow down a heart rate. All right, so that's just your basic overview of adenosine triphosphate. Your big takeaways are it's the energy currency in our body. If our body at the cellular level needs to use energy to get a process done, it uses ATP. All right. So what is the sodium potassium pump? It's basically, if you think about a cell, all right, there's channels, almost like doorways or windows into and out of that cell, because the cell membrane itself isn't 100% solid. It's got to allow things in. One of the things that we talk about a lot is the movement of electrolytes, sodium, potassium, and when we get into cardiac action potential in a little bit, we're going to be talking about calcium as well. It requires ATP in order to open that doorway. The doorway itself requires energy to get open, and that's where the phosphorus comes off of the ATP and opens that up. So let me just back out here real quick. I'm going to make sure my volume is up, which it was not. And we're going to go back to this video, and we'll skip some ads here in a second. All right.
Hi, it's Mr. Anderson, and in this video I'm going to talk about adenosine triphosphate, otherwise known as ATP. ATP is usually shown with this yellow starburst behind it, and that's because it contains energy. In fact, it's the energy coinage of all life on our planet. And most of you probably know that, but you might not know the structure of ATP. And so let's begin with the adenine. Adenine is a purine, and it's a nitrogenous base. That means that the black is going to be carbon, but the blue is going to be nitrogen. Where have we seen nitrogenous bases? Remember, that's going to be the rungs found on the inside of DNA. We also have a ribose sugar, and then we're going to have not one phosphate group. Phosphate is phosphorus surrounded by oxygen, but we're going to have 3 phosphate groups. And so let's start with the adenine and the ribose. They're attached together and they form a chemical called adenosine. If we add a phosphate to that, we have AMP, or adenosine monophosphate. If we were to add another phosphate to that, we've now got adenosine diphosphate, or ADP. But if we add that third phosphate, we now have ATP, or adenosine triphosphate. Now, these last phosphates are important. In fact, I like to think of the bond between these last 2 phosphates like a spring. As we attach that last phosphate on, we're storing energy, and so there's potential energy in that bond. There's also going to be potential energy in this bond right here as well. And so when we release that phosphate, we can release a little bit of energy. And so let's look at that. And so what is ATP? Here it is again. So we've got our adenine, our ribose sugar, and then we have our 3 phosphates. But you can think of it like a recharged battery. It's a battery that's charged up and has a certain amount of potential energy. In fact, if we were to take ATP, which would just look like salt if you were to have it in a solid form, and we were to add it to water, it would all hydrolyze. In other words, it would all break down into ADP. And so let's look at what's going on here. So if this is water, that water will break this last phosphate group. In other words, we're going to add an OH on one side and H on the other side. Remember, we call that hydrolysis, or breaking with water, and we create ADP. Now we have just 2 phosphates here. Well, what happens to that third phosphate? It's released. And so lots of times when you're dealing with ATP, you're going to see this P with a subscript of I. What does that stand for? That's this phosphate group that's been given off. And since there was a certain amount of potential energy in this bond, when it's released, that phosphate has a certain amount of potential energy as well. So in other words, if we were to let ATP just sit in water, it would all eventually hydrolyze and it would all become ADP, and we'd have these loose phosphates. That's going to be like a battery that's lost its charge. And so what do we do in life? Well, in life, we're going to tap some energy. And so we could do that in respiration by breaking down glucose, or in photosynthesis by taking in energy from the sun. And we're going to attach that phosphate group again. And that's called a dehydration reaction. And so when we attach the phosphate, we now make ATP. When we break it, then we're going to release energy. And so let's start by talking about the building. And so when do we produce ATP? We're going to do that in cellular respiration. And so remember, on the inside of that mitochondria, we have these proteins called ATP synthase. And what happens in all of respiration is we build up a gradient of protons on the outside of that, and as they flow in, we make ATP. Now, it's worth pausing for just a moment and talking about, how does ATP know where to go, and how does ADP know where to go? Well, if you're to think about it, let's look right here. As we're converting ADP and a phosphate back into ATP, they're disappearing. So you can think of, in the mitochondria, that's going to be the lowest amount of ADP and phosphate is going to be found right here. Why is that? It's because they're going to be converted into ATP. Well, where's the ATP going to flow from there? Well, diffusion is going to move it from an area of high concentration to low, and so it's going to move to wherever it's used. Once it's used, it converts back to ADP and a phosphate, and that's going to come right back here to the inside of the mitochondria. Now remember, plants are also going to produce ATP. They're going to do it on the thylakoid membrane, which is going to be this membrane right here on the inside of the chloroplast. What are they using to make ATP? They're using light, energy of light, to do that. How is this different than the mitochondria? Remember, the hydrogen protons are going to be on the inside of the thylakoid membrane. As they flow out, that's going to make ATP. What do we use that for in plants? Remember, out here in the Calvin cycle, we're going to use that to make sugars. Now, plants also have mitochondria, so they can break down that sugar and make ATP. And so that's how we're producing it. What does it do, though? Once we release that phosphate, what is it used for? Well, let me give you 3 good ways that it's used. So if we release that phosphate, it can be used in active transport. And so this right here is the sodium-potassium pump. This is going to be found on most of the cells in your body, but it's especially important in the neurons of your body because you maintain this gradient, and that's how...
I'm also going to interrupt him here. When we talk about the sodium-potassium pump and why that's so important is when we talk about cardiac muscle as well. We're going to talk about that in cardiac action potential, but also skeletal muscle as well.
Nerves actually work, or impulses work. And so let's watch what's going on. We have ATP right here. It's going to give up that phosphate, and that phosphate is going to bind to this sodium-potassium pump. As it does that, it's going to use the energy of that phosphate to pump 3 sodium ions out. We're then going to have 2 potassium ions that will bind to the inside of that pump, and that's going to flow to the inside as we release that phosphate. And so again, what are we using? We're using active transport to pump sodium out and potassium in. How much of your energy right now is going to that? About 20% of the energy in your body right now is going to ATP, which is running the sodium-potassium pump and keeping all those neurons working. So it's really important. How else do we use ATP? Well, when we release that phosphate, our muscles move using ATP. And so what's going to happen is the phosphate will bind to this myosin. And myosin is going to be a protein, and it'll attract that to an actin, and it's going to cause your muscle to contract. And so your muscles contract using this phosphate that is released from ADP. What do we have to do with that phosphate and ADP? Again, we have to regenerate ATP so that we can do this again. Where else do we have this? Well, in the formation of polymers. And so when we're building polymers, the easiest way to do that is to add a phosphate. And so let me...
All right, so that is the bulk of the information that I wanted you to get from that video. If you want to look at that video a little bit more, the link is right there. Okay, now let's talk about the cardiac action potential, and you'll see how this ATP and sodium potassium pump ropes right into this. Okay, so the cardiac action potential, couple things here. When we talk about action potential, this is basically the breakdown on how a muscle cell contracts and how it receives that signal, and the inflow and outflow of sodium, potassium, and calcium. In EMS and paramedicine, what we really deal with is a ventricular myocyte. So the waveform that I'm going to show you in a couple slides here is specific to the ventricular myocyte, the ventricular myocardium, that muscle cell itself. If we were to do an atria, or if we were to do skeletal muscle action potential, the waveform would be different, but the overall theory and process would be pretty much the same. Okay, so there's 5 phases of cardiac action potential. I'm not going to quiz you on this or test you on this right now. You're going to spend a lot of time in cardiology on this, but it's my job to give you the introduction right now of this. All right, so I'm going to bounce back and forth between these 2 slides quite a bit. So phase 4, right down here, and you'll notice that at the cellular membrane, we measure the millivolts, and we're at a negative 90 millivolts, and that's a normal resting state for a cardiac myocyte. All right, so the cells are at rest. There's no inflow or outflow of any electrolytes at this point. So this cardiac myocyte receives a signal, an electrical signal from the neuron that innervates at the neuromuscular junction from the nervous system into the muscle itself, and then from muscle cell to muscle cell. Okay, if you have ever taken an EKG class before, and you're all going to in your paramedic program, you'll know that the primary pacemaker of the cardiac muscle is the sinoatrial node, and that creates a signal about 60 to 100 times a minute at rest. So 60 to 100 times a minute, this cell, the specific cardiac myocyte, will receive a signal that says, okay, go ahead and depolarize. Okay, so depolarize creates a positive deflection of about positive 20 to positive 30 milliamps, and then the muscle cell will contract. So on this upslope on phase 0, the muscle cell receives the signal, uses ATP to start opening up those sodium and potassium channels, And your cardiac myocyte actually has fast-acting sodium channels and regular sodium channels. So on this initial upslope, you have a fast-acting sodium channel opening, and I'll draw this for you in class as well, and you get this rapid influx of sodium ions, okay? So as this happens, and as we peak here on phase 1, we get this rapid depolarization, okay? That is the electrical signal that your QRS is in an EKG. So you get that really big complex that's reading the electrical signal that's moving through there, and it pretty much corresponds with the depolarization here. So the sodium continues to come in, and potassium starts to move out of the cell. Excuse me there. Now, as the potassium continues to flow out, calcium actually enters the cell as well. So we've got potassium, which is your chief intracellular ion with your myocytes, flows out of the cell. Sodium and calcium are mainly in your serum outside. Extracellular ions, those will flow in, and then we cause a muscular contraction. That cell will actually contract. Phase 3 is when those ions start to reset, okay? And then the potassium outflow continues, and then eventually at the very end of phase 3, beginning of phase 4, everything gets reset, all right? And that process continues over and over and over, 60 to 100 times a minute. I know that's a difficult concept right now, and a very brief introduction. Like I said, submit your questions and tell me more specifically what information stuck for you, what didn't, and we're going to go much deeper into this in the lecture. All right.